Hello everyone. Welcome back to Law Scholar Independent Society. In today's biology class we will discuss about DNA and replication. But before this, kindly subscribe to our YouTube channel and also press the bell icon for more updates. So now let's begin the lecture. DNA is a molecule made up of two strands twisted around each other in a double helix shape. Each strand is made up of sequence 4 chemical bases represented as AGCT. The two strands are complementary. This means if there is T in a strand, then there will be A at opposite strand. And same as C and G. Each strand has a 5' end and 3' end and vice versa, run in opposite direction. The first step in DNA replication is a separation of two strands. And this unzipping is carried out by enzyme called helicase, which resulted in the formation of replication fork. Each separated strand act as the template or parent for the formation of new strand of DNA. An enzyme called primase starts the process. This enzyme makes the small piece of RNA called primer act as the starting point for the formation of new strand of DNA. An enzyme called DNA polymerase binds with the primer and will make new strands of the DNA. And this DNA polymerase can only add bases in one direction, from the 5' end to the 3' end. Now in the exercise, practice yourself about. What do you mean by bases? Define semi-conservative form of DNA. Define all the different type of enzyme used in replication with their action. Define replication. Now at the end of this lecture. If you like our lecture video, then like, share and subscribe to our channel. And hit the bell for more updates and stay tuned with us. Till then.